So I have 10 questions, and they're on your lab topic. So you'll recognize some related to absorption spectrum and some related to the RF factors and chromatography. So you might like a calculator. We're gonna do that. That'll probably take us about 10 minutes. And then I'm gonna give you a 20-minute break because I want you to make sense of what we've covered so far. And you might have already done this on your own, you might not have, but I think anything that is a process, it's very helpful to draw it out. So I want as much detail as you can put in that 20 minutes, label it, identify, you know, in a way that the light reactions make sense to you. So I don't want you to take this student's drawing. If we were in class, we would be drawing this on our tables. We would probably be doing animations and claymations as well. So I don't necessarily want you to take their picture or any picture from the internet and then just copy it. I want you to read through those steps of photosynthesis, and in your own mind, what does that look like to you? And I think that becomes very helpful when it comes to answering questions about photosynthesis, okay? So after our quiz, we're gonna take 20 minutes to do that. And then we're gonna come back and I'm gonna add on to what you already have. So I've given you a place to upload this in Google Classroom, and then we'll come back and we'll talk about the Calvin cycle. And then we'll finish our POGLs. So it's 7:50 right now. I'm gonna actually give you 10 extra minutes in case it takes you longer. So at 8:30, we are going to reconvene in a lecture sense and start lecturing, okay? So leave your videos on while you're taking your quiz. So everybody's gonna have their cameras on for the quiz. When the quiz is over, you can turn your camera off and you can walk away, or you can log off and then just log back on at 8:30. Okay, so we're gonna do it a little bit different today. What questions do you have about the timeline and what we're doing in this first half hour? So we're trying to make sense of photosynthesis by drawing it out. So including as much detail as we can, not necessarily copying. I don't want you to copy a picture. This is an example of what students in the past have done. So that sort of detail, making sense of all the steps of just the light dependent reactions, what we covered the other day. So then at 8:30, we're gonna start back up with class and we're gonna add content to that. Drawing this out, I think, is helpful because you have to be able to talk about photosynthesis in order to be able to draw it out, right? So if now I could take my picture and I can talk about sunlight is hitting photosystems and water is splitting into oxygen and hydrogen, and where does that hydrogen go? Oh, it's over here. So I see that it's being pumped through this electron transport chain. After electrons were excited, they move through the transport chain. Electrons were pumped into the thylakoid membrane, and then they diffuse out through the ATP synthase, which makes ATP. Other electrons are going to continue on to the second photosystem, which is photosystem 1, and they're going to get excited, and then they move through another electron transport chain where they go through NADP plus reductase and reduce NADP plus into NADPH, which will later be used in photosynthesis as well. So I think that that is super helpful in answering FRQ type questions, or any questions related to the light dependent reactions. These were two questions that a lot of people got wrong on the POGL. And I think this one especially. So a researcher is investigating the effects of a chemical blah, 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 permeable to hydrogen ions. Which of the following is most directly adding the effect of adding the chemical? So I'd want to know what the role of hydrogen is in this whole process, right? So if I go back to my picture, I'm able to see what am I doing with hydrogen. So after I split hydrogen, it was building up that concentration gradient in order to diffuse through and make ATP. So if you're, so the thylakoid normally isn't going to diffuse, right? Hydrogen in and out. It needed a pump and it needed a transport protein. So if it just became permeable to hydrogen, then you'd be losing hydrogen, right? So then you would lose your concentration gradient because diffusion would go to equilibrium. You'd lose your concentration gradient, and therefore you'd no longer be able to send hydrogen through ATP synthase in order to make ATP. So you kind of need to think about what the role of that particular item is in the whole process. And we know that it's related to ATP, so we really should be minimizing our thoughts to the ATP. This was another one that some people got wrong. And again, it has to do that with that proton gradient, and the proton is the hydrogen ions, right? So again, I need to limit my thoughts to things related to ATP, and there were 3 of these. And if which of the following statements provides evidence to support the claim that no ATP will be synthesized in the absence of the proton gradient? Okay. So if you took away that pump or you blocked those channels, you would no longer have a gradient, and so you'd no longer be able to make ATP. Anyways, I think your brain likes pictures, and so anything that is a process, that is something you can do, whether I tell you to do it or not, that is going to help you remember the steps, remember how things are related, right? And that's one of the things we're trying to work on is interconnectedness or relatedness of concepts. No better way to find an interconnection than to put them all on a graphic. Okay, so let's move on to our Kelvin cycle. Oh, I thought I had it open. Sorry, I thought I did have it open, but I think I closed everything because I got lost in my stuff. So today we want to talk about the light independent reactions. And why are they independent? Because they're taking energy from the first setup that we already did. So we're taking NADP and ATP, NADPH and ATP, in order to make organic molecules. I'm going to use this one. The student one, take it or leave it. It's there if you want to take notes along with me, or maybe it's a good thing to do after a lecture or while you're reviewing. See if you can go back and fill those blanks in when I'm not talking. Okay, so the light independent reaction is considered the synthesis phase because this is when we're synthesizing our organic molecules. And it's divided into 3 like separate phases, you could say. It's a metabolic pathway, so there's lots of steps along the way. We've talked about that. Every step requires an enzyme. There's an intermediate in between each of the things that we're showing you. And there's probably a few more steps than what we actually show. So we want to identify this as carbon fixation, reduction, and recycling or resynthesis of. So carbon fixation is the point when you're going to take carbon dioxide from the atmosphere, which the plants can't use directly to make organic molecules. So you're fixing it in a form that is usable by the plant. So I'm going to fix it into a form that we'll refer to as PGA. And you're going to see 2 different nomenclatures of this. So you'll see PGA or 3PG. And then another time you're going to see PGAL or G3P. So they're the same thing. Different resources will use a different nomenclature, okay? So we're going to take this 5-carbon sugar, which is ribulose. This is the one piece that stays in the cycle. So we start with it, and in the end, we make it again. So this is the cycling component, ribulose biphosphate, RUBP. CO2 comes in, binds with ribulose biphosphate, makes an unstable compound that splits in half into 3PG or PGA. How confusing is that, right? And then we're going to take ATP donate phosphates We're going to strip those phosphates off with hydrogen. And then in the end, we have this P-gal, or G3P. That is the 3-carbon compound that can be used to make other molecules. It can be used most commonly to make glucose. That's what we talk about. So I got to go around that cycle twice. But it can also be used to make the glycerol of a triglyceride. It can also be used to make amino acids. So we make lots of different organic molecules, or we don't, but plants do, from that P-gal. And then we're going to recycle the RUBP so that this whole process can go again. So you should be able to talk about that. So like I just took a picture and I explained the picture, okay? That's how pictures can help you with learning material or understanding material. Draw it out yourself and then talk about your picture. So we definitely want to see the connection between light and dependent and light independent reactions, which is real quick and easy, NADPH and ATP. Repeat the steps of Calvin cycle like I just did. Draw it out when you're done. Identify NADPH and ATP. What are we doing with them that allows us to convert CO2 into glucose? And then I don't know that I want to hit photorespiration today. Down the road, we're going to identify adaptations that plants use so that they're not using the oxygen that they're making or that they're bringing in. They're not using it for respiration, but they're actually using it for, I'm not saying that right. RUBP can bind with O2 or CO2. And so the O2 that you're making, you don't want to bind with RUBP, or you won't be able to make glucose. So that's another day because it gets confusing if you talk about 2 topics at once. Okay, so we want to look at our chloroplast as a whole, right? So what we've been doing so far is taking place right here in the grana. In each of those individual thylakoids, that's where the light-dependent reactions occurred. So now we're going to be outside of the grana in this open space. A lot of people labeled the stroma wrong on your interactive worksheet. So the space within the chloroplast is the stroma. You might think of it as like the cytoplasm of the chloroplast. Remember, this was probably once a bacterium. Okay, so that's where this part of photosynthesis is occurring. We have to have ATP and NADPH. That is why we need the light-dependent reactions to occur. In the first, there's 2 different pathways, and we're using C3 today, okay? C4, I'm going to say for another day. It is that adaptation that allows plants to not respire the oxygen that they're using. Okay, so carbon fixation, taking carbon, making it into a usable form like glucose. CO2 by itself, very little oxygen when it's fully oxidized. So we might remember oxidation reduction, right? So oxidation is gaining, it's losing electrons. Lose electrons, you're oxidized, and gaining electrons, you're reduced. So usually the more complex molecule is the reduced form because electrons take place in that bond, right? So CO2 doesn't have a lot of energy, just the carbon-oxygen bond, not a lot. We learned that carbon-carbon bonds have a lot more energy. So, so to go from CO2 with not a lot of energy to glucose, which has a lot of energy, that is going to be an endergonic reaction, right? We have to climb a big hill to get past our activation in order to store that energy. And that's why we need the light energy from the sun. Okay, so then we're going to reduce CO2 step by step into glucose. Again, every step is an enzyme. Every step could be messed up if that enzyme was malfunctioning. So this is the big picture. Try to look at it in 3 steps, remember, carbon fixation, reduction, and recycling. So you're going to use 9 ATP, 6 here, 3 here, along the way. You're going to use 6 NADPH. So notice right away we need more ATP than we do NADPH. And that's going to play a role in cyclic respiration or cyclic photosynthesis. 3 molecules of CO2 are going to be fixed, turned into a usable form, into glyceraldehyde. 3-phosphate, that's your G3P. Okay, so once I add this ATP, the ATP is going to add a phosphate and put this in a more active form in order to bind to the next enzyme. So that's basically its purpose. So, so we're going to make phosphoglycerate, which is your PGA. We're going to convert it into phosphoglyceraldehyde. That's this end product. That's that G3P. Okay, so this G3P is all the way here to the end. So step by step, we'll get there. Through some vocabulary makes it easier to talk about. The Calvin cycle, also called the light independent reactions. We talked about Melvin Calvin already. RUBP is short for ribulose biphosphate. That's the 5-carbon sugar that stays in the cycle. Okay, you should be familiar with RUBP. Rubisco is an enzyme, and we need to be familiar with that enzyme. So it's ribulose biphosphate carboxylase. So ASE makes it an enzyme, right? RUBP tells you that's one of its substrates. What's its other substrate? Carbon dioxide. So it's going to combine those two together. PGA is phosphoglycerate, and it's also called 3PG. So this is that different nomenclature, trying to give you something to keep track of it. PGAL phosphoglyceraldehyde is also called G3P. So these words might be used interchangeable. I have a tendency to use PGA and PGAL. But I'm trying to use both when I talk to you guys. So carbon fixation, the first phase, is taking CO2 and turning it into that PGA. Okay, so with the help of the enzyme Rubisco, these two will bind together. A 5-carbon RUBP plus a 1-carbon CO2 makes a 6-carbon compound. That 6-carbon compound is unstable. It immediately splits into 2 3-carbon compounds. Those 3-carbon compounds are your PGA. So then I'm going to go into my next phase, the reduction phase, where I can add electrons. I'm adding electrons via the NADPH. Remember, NADPH is an electron acceptor. That means it can move the electrons from one reaction to another. So it's going to take electrons and donate them to the PGA, reducing, you see, because you added electrons, reducing the PGA into another form, which is PGAL. Okay, this happens in 2 steps. It first requires ATP. ATP donates a phosphate, puts it in its active form to be converted into PGAL with the help of that electron. That's the second phase. The third phase, this PGAL is going to go off to be used to make organic molecules. The rest of it, so I have 5 carbons left. The rest of it is going to go back to make RUBP. So it rearranges those molecules in some unknown way. Somebody knows it, but we just skip right ahead to, it's going to take ATP in order to do that. And then we repeat it, and I will play that song another time. So this is the big picture again. CO2 is coming in, binding with the enzyme Rubisco and RUBP, forms PGA, phosphoglycerate. That will take a phosphate, which will be replaced with hydrogen and electrons to form PGAL. PGAL is used, and the rest is recycled. You're doing this with 3 CO2s at the same time. So it's all these numbers times 3, okay? So you have 3 CO2s coming in, binding with 3 RUBPs. That makes 3 6-carbon compounds that are unstable and split into 2 3 carbons. So now I have 3 times 2. I have 6 3-carbon compounds. It's helpful to move little things around to help you see where these carbons are going. Adds a phosphate to each of those 6 PGAs. The phosphate is stripped off by hydrogen, reducing the PGA into PGAL. So now I have 6 PGALs. Only one of those 6 goes to make organic molecules. So from 18 carbons, I went to 15 carbons. I need to make 5 carbon compounds. So with 15 carbons, I can make 3 of them. So I'm back to the 3 original RUBPs. So hopefully this will open. I think this animation is super helpful in picturing what's going on. Okay, so this is my enzyme Rubisco. This is my 5-carbon RUBP. My CO2, which is unusable until I fix it, right? So in the first phase, I'm going to fix CO2. So you're noticing CO2 binding with RUBP and it immediately splits. So it doesn't spend any time in that 6-carbon compound because it's unstable. So here's my ATP and my NADPH. ATP, watch for it to add a phosphate. Watch for NADPH to replace that phosphate, okay? So phase 2 is reduction. You're adding electrons. ATP adds a phosphate. NADPH removes it. What it's actually doing is adding the hydrogen and electron, which cause the phosphate to break off. So of those 6, they were all converted into PGAL or G3P. 1 goes to make organic molecules. The rest of these are going to be used to recycle RUBP. ATP has been converted into ADP. We donated a phosphate. NADPH has been converted into NADP+. They are sent back to the light reactions to do it again, okay? So in the third section, we're going to recycle these 5 PGALs into RUBP, just a whole lot of letterings. And this is the magic. I say we don't really know the steps that happen in there. We don't need to know them, but we do need to know that it requires 3 ATP to reform. So they're going to donate phosphates to reform that RUBP. Okay, so click on that animation later to review that. I think it's a super helpful animation for seeing what's going on. So in the end, we took ATP and NADPH from the light reactions here in the thylakoid. We consumed water, replaced hydrogens, lost oxygen. That oxygen is a byproduct. We breathe it in, they respire it. Calvin cycle, so NADPH and ATP are going into the Calvin cycle. They're used to reduce CO2. So CO2 is fixed and then reduced into PGAL. And at the same time, they're regenerating ADP and NADPH so it can go back into the light reactions. So that's a lot of complicated words. This is where drawing things out is helpful. Make your own flow chart. So take these steps and do whatever it is you need to do to make sense of them. Maybe it's a flow chart. Maybe you're drawing out all of your carbons and seeing where the carbons go. Basically, you need to know what goes in and what comes out. Okay, you don't necessarily need to know how one molecule is converted into another.